Okay, you're muted. Whenever you're ready, you can take control.
Okay, hello. Can everybody hear me?
Yes.
And see my screen? Okay. So my name is Amelia Faust, and I was in the Monarch Meadows research group this semester with my mentor, Gail Tompkins. So for our project, our main goals were to build habitats for monarch butterflies, to encourage biodiversity in our experimental fields, because biodiversity leads to healthy ecosystem, and to develop environmental awareness. So monarch butterflies make this giant migration from Mexico up to the northern part of America and the southern part of Canada every year, and they do this over the course of 4 generations. So generation 0 in the spring will wake up from their hibernation, and they will fly to northern Mexico, where they will mate, they will lay their eggs on milkweed, and then they die. And then their offspring makes up generation 1, where they fly even further north and again mate, lay eggs on milkweed, and die. Their offspring, generation 2, flies slightly further north and mates, lays eggs on milkweed, and dies. And then their offspring, generation 3, will have reached as far north as the monarchs travel, and they again will mate, lay eggs on milkweed, and die. And by the time we get to the 4th generation, it's about late summertime. So instead of mating, these monarchs will feed on the late blooming flowers, drink their nectar so they have enough fuel to make the long journey all the way back to Mexico, where they hibernate through the winter, and they become generation 0 in the next cycle. So monarch butterflies don't actually make it to North Carolina until mid to late summer, so we spent this semester preparing for them. I have a couple example photos here of things that we planted, because you'll see in a minute our progress photos are still developing. So on the right there is an example of milkweed that we have planted in our flower beds. And this is vital for the monarchs because milkweed is the only plant on which they lay their eggs. And once the eggs hatch, their caterpillars eat the milkweed. And then they attach their chrysalises on the milkweed. So the milkweed is pivotal for these monarchs' whole life cycle. And then on the left are coneflowers, which we planted in our experimental field. So the monarchs have nectar to drink. So again, on the left there is an example of one of the coneflowers we planted in our field, and we're hoping it's going to take root and become a vital part of our little ecosystem in our small field. And then on the right there is one of our flower beds that we have growing in it milkweed. We have some bee balm, and we have other plants that monarchs really like. So we are optimistic as we continue our work in the summer that we have made a nice habitat where we can study our monarchs. That's about all I got.
Thank you so much. Does anyone have any questions?
I'm saying our summer and fall activities are really where the research and the data comes from. Spring is all about maintaining the habitat, and our students have been fabulous with that.
Yeah, we're excited. I know our other person in this group is doing some research with like how a certain parasite interacts with these monarchs. And I'm looking to see more like how they interact with the actual plants, types of milkweed, or, you know, if they prefer younger or older leaves to lay their eggs on. So we don't have much information about any of that yet, but we're excited to keep working on it.
That's great. Thank you for sharing. It looks like there might be another message coming through.
So we are trying really, monarch populations have been dwindling for a while. So it is really important for everybody to do what they can locally, like in their garden, in your local community, to plant, milkweed and pollinator flowers. So the monarchs can keep their population numbers high, because there's a lot of problems with deforestation down in Mexico that is really hurting their populations. There's a lot of problems with dwindling flower numbers in the late summer, so they don't have enough food to make it back to Mexico. So our research, we're trying to make sure their short little time here in North Carolina are good, and they can be as strong as possible, so they have the best chance of making it to Mexico and continuing the cycle every year, because we don't want our monarchs to die out. So that's the main goal of our research.
Thank you. Good answer. I think there's maybe one more question coming in, so I'll give it just a second. Maybe not. Okay. All right. If you do have any other questions, feel free to put them in the chat, and we'll come back to these at the end after our other presentations. So thank you so much.
Thank you.
Let's see. Next up, we have Ananya Jain. And there she is.
Okay. Hi, everyone. I'm Ananya Jain, and my START project this semester was aquatic flora and fauna. So basically, the goal of my project was to find the difference between macroinvertebrates found in upstream versus downstream areas. So our study site was South Campus Stream, and if you look there, that orange dot is exactly where we are. And the reason why we kind of chose this site was because there was a bridge located in this stream. Now, this bridge served as kind of the disturbance that we wanted to see, and we wanted to investigate whether it controlled the different kind of macroinvertebrates found between upstream and downstream, as shown here. So this is the picture of the bridge in our real time, and then we have us at upstream and downstream areas of the stream too. So the method that we used was kickneck sampling, and that's shown in the picture here. And that's the net also shown there. So with the sampling, we did 3 rounds of sampling across the stream, and we basically kicked a bunch of the rocks, scooped up the nets, and saw if we found any macroinvertebrates caught in our nets. So now, before talking about what we found, I just kind of wanted to explain how to understand it. So each macroinvertebrate is given a biotic index. Now, depending on this biotic index, it tells us about how good water quality is in the stream. Now, a score of 0 means that they are not very tolerant to water pollution, where a score of 10 means that the macroinvertebrates are tolerant to pollution. So ideally, we would want to score closer to 0, indicating positive water quality. So these were the species that we found. So we went out twice, and on the first date, we found two macroinvertebrates, and on the second day we found none downstream and we found 2 macrovertebrates upstream. So downstream, first of all, we found a leopard frog. Now, this is actually a vertebrate. I just wanted to include it. And leopard frogs have a bioindex close to 0, and there's that. Then we also found crayfish. The crayfish for bioindex is unavailable. And then we also found a damsel nymph, and that has a bioindex close to 0. And then we also found a salamander, which also has a bioindex close to 0. So the conclusion was that through START, I was able to learn about experimental design. That was my goal going into it. And I was able to witness the connection between planning a study and achieving results. Through this study, I actually found that the disturbance of the bridge actually did not affect water quality that much as I thought it would. And just on a broader scale, as a high school student in START, I also learned that observatory science is just as important as experimental science, and that any research that I do requires a significant time commitment and a passion for research. And START has taught me skills that I'm looking forward to taking in future research. And aquatic flora and fauna has taught me much about our environment, and I'm glad for that. And that's all I have.
Thanks, Ananya. Anybody have any questions?
When did you do your sampling?
So we went out on, so I actually wasn't present for both samplings because of my high school schedule, but the times that my rest of my team went out was on March 19th and then also April, I think I want to say it was, sorry, it was March 19th and April 6th, yes. And both times we measured upstream and downstream.
Okay, we've got a lot of comments about how interesting it sounds. I liked your comment about how observational research is important too. So thank you for that. Any other questions?
I think the replications of our study are possible. I didn't go as in depth into it, but for kicknet sampling, we were able to do the thing where we reached our site, we measured 100 meters from our disturbance, both upstream and downstream, and then repeated the kicknet sampling 3 times in the exact spot on both different occasions that we went. So because of this, I feel like replicating the study would be possible, and that's why the results of the study are able to be conclusive.
All right, thank you so much.
Thank you.
Shrija is having some internet connection issues. We're going to try to get her on later. So we're going to skip ahead a little bit and have Caleb Holder do his presentation now. Caleb?
Sorry about that. I forgot to unmute.
No problem.
So hello, everyone. Thank you all for coming to the START Symposium. My name is Caleb Holder, and I'm part of the aquatic flora and fauna team. My goal was to address the soil as part of a stream ecosystem and to learn how it could potentially be affecting this stream in specific through erosion. When beginning our study, I had observed that the land maintenance practices affected the turbidity in a stream near my house. And from this, I predicted that if the soil particles are able to make their way into a stream, pollutants suspended in those soils could be present in this water as well. And so by testing the soil for pollutants and aggregate stability, I could infer about both the presence of potential pollutants in the soil and the potential for those pollutants to enter the stream through erosion. On Southern Campus, there was a bridge that was built in this location circled in red, and then this next photo is the bridge itself. And so we were hypothesizing that this interference could be affecting the stream. So I decided to do a transect sampling style to allow for a uniform collection of these samples. And these samples were taken 30.48 meters both up and down the stream of the disturbance and 8 meters off the bank in both north and south direction, with the stream running west to east. 1.5 cups of soil were taken from 15.24 centimeters beneath the soil, both up and downstream, to be sent for chemical analysis at Brookside Labs. Another 4 samples were taken, these being only 45 milliliters of soil, and they were going to be at the same spot, the same depth, and were used for aggregate testing. And this was repeated twice about 3 weeks apart. So I decided to use the SLAKES application as part of my aggregate testing, which compares with the area of the aggregate before and water is added to determine the soil's ability to withstand erosion and harbor biological life. The collected samples needed to dry for at least a week, which is that bottom picture, until there was no water left. But as I was drying the samples inside my house, which is usually kept at 20.5 degrees Celsius, I let the samples dry for at least a week and a half. Using the SLAKES application, as seen as the top photo, on my iPhone 12, I collected 4 pea-sized aggregates and weighed each one to have as a record. I then followed the instructions on the SLAKES application and recorded the results and the math done within the application. What I found was that the soil I collected was considered a stable soil, and my results below 2, meaning that this soil was less likely to succumb to erosion and be adequate for sustaining biological life. The number from the SLAKES application has a negative correlation with soil health, meaning that the better the soil health, the lower the number. This chemical analysis, as of now, is being returned from Brookside Labs. We have just sent it out just recently. And so a comment that I have on the SLAKES application was, in order for the app to work, very specific needs needed to be met, which of some were uncontrollable, such as the aggregate falling apart. But the main controllable factor in this aspect was the light and trying to make sure that the shadows were decreased to make sure that the application was able to recognize the aggregates. And so this is my data here. And so that top slate result number was talking about the output. And so here I have the water as well as the aggregate weight. And so the aggregate weight was not really a necessary portion in this experiment, but I just wanted to include it just to have as a marker. And so this is the works cited as for the SLAKES application. And so as a team, we observed at our site that the turbidity of the water changed dramatically between the day after rain and multiple days after rain. So a potential question we were kind of looking at was going forward was, where are these particles originating from if it's not that land stream, you know? And so the Highway 540 being built just above here, we had a once healthy stream flowing straight through like a tube. And so going forward, we kind of want to address that to see if that may be affecting our results. Yeah, so that's pretty much that.
Well, thank you, Caleb. Any questions for Caleb? No questions coming in yet, just lots of praise. Okay, if we don't have any questions, or if you think of any in the meantime, go ahead and put them in the chat. We were able to get Shrija to join on her phone, so we're gonna try to give it a try. I'm gonna share her presentation, and we're just gonna make the best of it and see how it goes. So no more questions for Caleb. Thank you, Caleb. We will open Shrija Nupani's presentation, and then she will tell us a little bit about it. So give me 1 second to share. And I will turn off my camera. Shrija, are you there still?
Yes, I'm here. Can you hear me?
Okay. Yes, we can hear you. All right.
Hello. Should I start now?
Yes, go ahead.
Hello, everyone. My name is Shrija Nupane, and my START project was aquatic flora and fauna. The part I'm focusing on is invasive plant species. Next slide, please. So the goal of my research is to see if there are any differences in the diversity of invasive and native plant species when there is human intervention involved. These are some pictures of me in the field. Next slide, please. So organisms that damage ecosystems in which they do not occur naturally are called invasive, alien, or non-native species. These species are usually brought into new habitats to control native species, other existing invasive species, or by pure negligence. Invasive species tend to outcompete existing species by taking over the existing habitats. These actions tend to threaten the balance of fragile ecosystems. The two main lead causes of species extension in their natural habitats are invasive species and human intervention. So these pictures are some of the common invasive species I found in my observation. Next slide, please. The site chosen to collect our data sampling was around the new construction being done at Wake Tech Community College Southern Campus. My prediction is that there will be more invasive species downstream versus upstream due to human intervention within the natural habitat. The methods of sampling. So in order to find my data points, I traveled about 30 meters from the disturbance upstream and downstream and created a secondary transect in which I created quadrants at 9 meters and 18 meters perpendicular and away from the stream on both sides. This enabled me to have 4 quadrants upstream and 4 quadrants downstream. In order to acquire a more diverse data set, I collected data over 2 different days. The method I used to get my sampling was by creating a quadrant with 4 1-meter sticks. To avoid bias, I threw a meter stick behind me and created the quadrant to the right for all 16 quadrants. Within each quadrant, I looked for different species of plants and the percentage coverage of each species on the ground by glance. Next slide, please. So this is the table of average plant coverage. According to this table, there is a prevalence of Japanese honeysuckles, Chinese privets, and creeping jenny in my observation sites. All 3 of these invasive plants are highly persistent, hence such high percentage of coverage. Next slide, please. I created an Excel graph with the data set in order to discover the percentage of coverage each plant had within a quadrant. This was done to see whether or not there were more native plants versus invasive plants. In the following slide, I will show the average percentage coverage in the quadrants, upstream in red, downstream in blue for each of the species. So at the bottom, I don't know if you can read it, but those are all the species I observed. Next slide, please. As you can see in the graph, the species found upstream away from the construction flow has more varieties of species compared to downstream. These are all the species compositions that were sampled. Next slide. Based on the graph, an abundant amount of Chinese privets were seen during this observation. Chinese privets are invasive species that originated from China. Chinese privets tend to suffocate the native species nearby and take away the nutrients. This caused these native species to die quickly and the Chinese privets to multiply. Next slide. The data collected from both days showed that there are more invasive species upstream and more native species downstream. The data also shows that there is more ground coverage downstream overall versus upstream. This essentially means that even though there were more plants downstream, most of these plants were native species. Next slide. The conclusion. My original prediction was wrong. In fact, what the data showed was the reverse of what I had expected. There were more varieties of invasive and native species upstream away from the construction flow versus downstream towards the construction flow. A conclusion that can be drawn from this experiment is that due to the construction, there may have been a reset on the biodiversity life downstream. In order to rebuild itself, the land downstream was able to initiate a fresh start. So these are some more pictures of the site. Thank you very much.
All right, thank you very much, Rachel. Does anybody have any questions?
Thank you. Thank you.
There is a question in the chat. Jessica asked, what methods did you use for identifying the plant species?
Oh, so for the identification, I used a plant called Picture. I used an app called Picture This. So I would take the picture of each leaf that I found within a quadrant, and they would tell me which one it is. And in order to make sure that that was the plant, I also used the app iNaturalist to cross-check. Thank you.
All right, very good. Thank you so much. We've got a couple minutes left. If anybody has any other questions, please speak up or enter them in the chat. I am sharing a couple of links for you if you're interested in checking those out. These are for the Futures program, the NC Science Festival, and our START program here at Wake Tech. Any other questions? Luke.
Hey, yeah, it's not so much a question than a plug. All the research that's done at Wake Tech, you can, if you're interested in seeing it in a little bit further down the way, you can check out Eureka. It's a scientific publication by Wake Tech of the research done at Wake Tech. So any student in this session who is interested in getting some publishing experience can try to can work towards publication of their research that they've done with START at Eureka. And I'm not just saying that because I'm an editor there. It's really a great resource for you to get more experience with science. So just wanted to mention Eureka.
Yes, thank you for sharing. We'll try to find if you have a link to that, feel free to throw it in the comments for us so that everyone can check it out. And thank you everybody else for joining. It was a really interesting session from the students. There, he did share a link with us. Thank you, Luke, very much. Students, check it out. All right, thank you, everybody. I'm going to end the recording now. Have a nice day.